In this video, we're going to look at a very simple tool that allows us to see all the possible random fertilization events for any given genetic trait. To give you a bit of the background, there was a plant scientist named Gregor Mendel. He conducted an investigation on pea plants and collected a lot of data in order to provide evidence for the mechanisms of heredity. Mendel worked on the experiment because he was interested in the laws of heredity, how heritable traits such as the color and the shape of the pea plants were transmitted from the parents to the offsprings. At first, his work on the pea plants was essentially ignored. But long story short, his experiment was later rediscovered and his laws of heredity were accepted as truly fundamental to all living things. So after the rediscovery of Mendel's work, there comes along another scientist named Reginald Punnett, where he derived Mendel's ideas and laws of heredity and created the simple tool called Punnett square. What happens here is, again, the Punnett square is a tool that gives us the visual of what's going on in the Mendel's experiment. And another feature of the tool is that it displays all the possible random fertilization events. I'll show you what I mean. So here is what the Punnett square looks like. And this particular Punnett square is for the monohybrid cross. It's very simple. So because this is a monohybrid cross, we're going to have only two genotypes that are going to be crossed. Or another term is hybridization. The two genotypes are going to be hybridized with one another. Let's say our first genotype is in the form of big A and small a. Let's make this the paternal. Our second genotype is let's say it's big A and small a also. So this gotta be the maternal. Before we start filling out the empty boxes, I think it's important to bring up one of Mendel's laws. The law states that genes exist in pairs. Well, what does that mean? So we understand that our genes are what makes us unique, right? Genes control our traits. When we say genes come in pairs, what that means is a gene has two alleles, and those two alleles may come in different forms. And the different forms of the alleles will give off different traits or features in an organism. So for example, this gene has the genotype allele big A and allele small a. And let's say this gene controls our height. Although height is controlled by many different genes, to make it simple, let's just pretend this one gene is in control of our height. So this height gene from the mother here has the genotype of allele big A and allele small a. Now let's fill in the empty boxes already. So what we see here is a total of 4 genotypes. A homozygous dominant here, because it's big A's, and another homozygous here. But the letters are in small case, so we call this particular genotype homozygous recessive. These twos are the same thing, one here and the other here. They are the same, so they are heterozygous with the dominant allele as the capital letter A and the recessive allele in the small case letter. Each offspring that is going to be born is going to have 1 in 4 chances or 25% chance of being big A, 25% chance of being small a, and 50% chance of being big A and small a.